The human genome contains 3164.7 million bases. Every gene consists of 3000 bases. There is one largest human gene, that is dystrophin, 2.4 million bases, and the almost all 99% nucleotide bases are exactly the same in all people, and the functions are unknown for over 50% of discovered genes. The less than 2% of genome codes for proteins. Repeated sequence make large portion of human genome. Then repetitive sequences, the stretches of DNA sequence that repeated many times, and they are thought to have no direct coding functions, but they shed light on chromosome structure dynamics evolution. Chromosome 1 most genes 2968, and Y has the fewest one, 231. Then the 1.4 million locations where the SNPs, single base DNA differences, SNP single nucleotide polymorphism, and this also in the use of disease and tracing human history, which is simply sequencing the whole set of genome that contain all the coding and non-coding sequence, later assigning the different regions in the sequence with the functions. This known as the sequence annotation. And chromosome 1 was completed only in May 206. It was the 24th human chromosomes, that is 22 autosomes X and Y. This one is the incomplete dominance example. Capital B represents the round starch grain and round starch grain, and then small b represents the wrinkled one, and capital B small b, these are the intermediate size. And the 5' to 3' end, this one is the coding strand, and the promoter at the 5' end of the upstream. The 5' end of the upstream, that is the promoter, and the 3' end towards the downstream. Upstream and downstream, the 3' end of the coding strand is the terminator. The T cells themselves do not secrete antibodies but help B cells to produce them. So the several genes called the cellular oncogenes, C-onco or proto-oncogenes, they found in normal cells, which when activated under the certain conditions could lead to the oncogenic transformation of the cell.